The overall goal of this protocol is to perform high-precision inter-dye distance measurements using multi-parameter fluorescence detection of fierce-to-resonance energy transfer.
This method can help answer key questions about biomolecule functions by identifying conformational states and investigating the functional relevance of those conformations.
The main advantages of these techniques are its precise determination of inter-dye distances and that it corrects for experimental artifacts commonly observed in single-molecule FRET experiments.
The implications of this technique extend towards determining structural models of biomolecules using FRET-derived distances because it allows identification of conformational changes occurring in the sub-millisecond timeframe.
Generally, individuals new to this method will struggle with the analysis because of the large set of parameters that are determined in the experiment.
Demonstrating the procedure will be Junyan, a grad student from our laboratory.
To begin the experiment preparation, suspend 0.1 grams of charcoal in 15 milliliters of sterile phosphate-buffered saline. Pass the mixture through a 0.2 micron syringe filter. Store the charcoal-treated PBS in a sealed container at room temperature. Next, add 500 microliters of distilled water and 5 microliters of polysorbate 20 to the wells of a chambered cover glass. Mix the polysorbate 20 solutions thoroughly and allow the solutions to sit in the wells for 30 minutes. Then remove the polysorbate 20 solutions and rinse the wells twice with distilled water. Allow the chambers to air dry. Next, in a microcentric